Hi, everyone. This recording is going to be on protein metabolism. This is a really interesting topic as I profoundly, investigated deep into it. So, we have 20 amino acids. Okay, remember, the first thing to remember is that proteins are the most abundant and most important organic molecules. That's the first step. Second, proteins, the building blocks of proteins are amino acids. There are 20 amino acids. 10 of them can be considered essential, okay, meaning that you must obtain from your diet. There is a couple of them that we can make them, but they can be deficient in a specific times in our lives. So we'll talk a little bit about that. So in general, considering those specific events, we usually refer to 10 of them being essential, okay, because of that. Now, in here, we have the 20 amino acids. Okay, so let me scroll down. We can see that of the 20 amino acids, 18 of them are glucogenic, and 2 of them are ketogenic, meaning that they can be used to make ketone bodies. Okay. Now, you make ketone bodies when you're fasting. Especially this is, you heard about ketonuria in diabetes type 1. That's poorly managed, diabetes. So if the diabetes is not well managed, then the body will not be able to drive in glucose into the cell, and it will use other sources, to make to make energy. It will use other sources to make energy, like fatty acids and amino acids in this case. Now, the 18 amino acids, 13 of them are strictly glucogenic, and 5 of them can be glucogenic and ketogenic. Okay, there is a mnemonic. I'm not going to ask you about this, but this mnemonic helps us to remember which amino acids are both glucogenic and ketogenic. Okay. All right. So glucogenic, meaning that we can make glucose from them. Okay, and this is an important aspect of this presentation. Okay, so in protein metabolism, this takes place, mainly in the liver and kidneys. Okay, and remember that metabolism is the sum of the anabolic and catabolic processes that take place in our body. So now we're going to talk specifically here of amino acid catabolism. Okay. In amino acid catabolism, you can see that a carbon skeleton is used to generate energy, and an amino group is transferred. Okay, it's transferred. All right, so this transfer of this amino group is essential to make those amino acids that are made by the body and not obtained from the diet. Okay, so we have those amino acids that are essential. You get them from your diet. You obtain them from your diet. But there's amino acids that your body makes. Your body makes. Okay, and those amino acids are called non-essential amino acids. So those amino acids that are non-essential, that you cannot get from your diet, your body is making them through this process here. Okay, so this is why this is important. These transamination reactions. This first sentence here is pertaining to these transamination reactions and even the amination. But in the amination reactions, we get something else as well, which is, the removal of, the formation of ammonia. Okay. The formation of ammonia, and ammonia is a toxic chemical is toxic to your brain. Okay, so it needs to be, converted to another chemical that is less toxic. Okay, and this is called urea. So ammonia needs to be converted to urea. So we have in the liver, we have the urea cycle, okay, in which we're converting ammonia to urea, and urea can safely be, released into the blood, and the kidneys will remove urea. Urea can also be reabsorbed by the kidneys, to help us retain water, okay? But it is a waste product. Okay, now for both of these reactions, there is a vitamin that is essential, that is needed, is required. Okay, and so it requires a coenzyme derivative of vitamin B6. Vitamin B6 is known as pyridoxine. So this coenzyme that's a derivative of vitamin B6 is called pyridoxal phosphate. We need to know that. We need to know that this coenzyme is required for these processes here. Okay, now let's talk about transamination. Okay, so here's the image that the book provided us. Okay, we have glutamic acid, which we can call glutamate. We have a keto acid, number 1. And then on the other side, notice that this reaction is reversible, okay? And this reaction here is being carried out, by these transaminases. The name of the transaminase, usually it has to do with one of the reactants here. Like, for example, glutamic acid, we have glutamate transaminase, okay? And then on the other side here, we have a keto acid, number 2, and a new amino acid, which is known as tyrosine. Tyrosine happens to be a non-essential amino acid, okay? It is made from, well, I don't want to get into the details, okay? Because there is different amino acids can be made by different by different things, So I don't want to complicate things here. But in here, in transamination, what we have is the transfer, this transaminase is transferring an amino group with this hydrogen atom here, okay, to a keto acid, right? To a keto acid. And then that keto acid will be now a new amino acid, okay? So we're transferring an amino group to make an amino acid, which is usually an amino acid that we don't get from our diet, that the body makes it. The remaining, which is the carbon skeleton here, right? The remaining of it will be the keto acid number 2. And that keto acid number 2, even though they're not showing you the name, can be used, for energy. Okay, it can be used for energy. Okay, and so because this is so superficial, and that's really what you want to know for the exam. Okay, that in transamination reactions, an amino group is transferred to a keto acid, and a new amino acid is made, and that's it. They don't tell you anything else. Okay, I want to tell you a little bit more. Okay, because this is so important. The purpose of learning this information is to see how it can be applied. Okay, so I went digging deep because I have curiosity, I have curiosity. And you can see here, when you read it, it says in transamination reactions, which is a reversible reaction, it attaches an amino group of an amino acid to a keto acid, converts the keto acid into an amino acid that leaves the mitochondrion and enters the cytosol. And that amino acid is going to be available for protein synthesis. But notice I didn't tell you the other part, which is what happens to this keto acid too. So this one here can be used to make energy. And I'm going to show you that now, okay, because this is important for gluconeogenesis. Remember that gluconeogenesis is making glucose from non-carbohydrate substrates, okay, like amino acids and fatty acids. So we can make glucose from this non-carbohydrate substrate, okay? So let's see here. So here's some transamination reactions here. And the reason I picked these two here, because they're the most important. You know, when we do a liver function test, we're testing for specific transaminases, okay? And if they're elevated, that is indicative of liver disease or there is damage to the liver, okay? I put that stuff right under here in blue, okay? But here's alanine. Alanine is the main amino acid that is coming from muscle, okay? And at the end of this presentation here, at the end of these notes, you can see an image of that, okay? You can see an image of that. But alanine, okay, you can see the amino group here, okay? They're putting emphasis on that, okay? You know, the hydrogen atom is here with the carbon, with the central carbon here. Okay, this happens to be a chiral carbon. You know, it has like 4 different things attached to it, okay? Now, alanine, notice that the amino group from alanine is transferred to an alpha glutarate. To do that, we need paradoxal phosphate. Remember that was the coenzyme that was required. Okay, so paradoxal phosphate is required. Okay, you can see that is first transferred to paradoxal phosphate, and this transfer is mediated by alanine transferase, aminotransferase or alanine transferase. Now this product here is very small here, the name here, this is called paradoxamine phosphate. Now, and notice that again, you know, this alanine transferase here now transfers this amino group to alpha ketoglutarate, okay, and now it becomes glutamate. It becomes glutamate. So once it's given to alpha glutarate, it becomes glutamate, okay? And glutamate is another, you know, amino acid. Glutamate is an excitatory amino acid. You know, it's the most important excitatory neurotransmitter in the brain, glutamate. The other one is aspartate. You see it here. Okay, but you can see that, you know, so you can see that the amino group was transferred to a keto acid. Here is keto acid number 1. And then, and this is, you know, here's keto acid number 2, by the way, the one that is keeping the hydrocarbon chain here. I mean, the carbon backbone here. Okay, so, and what is this keto acid number 2? This is pyruvate. Look at that, pyruvate. From pyruvate, we can make glucose. Okay, so pyruvate is a valuable precursor for gluconeogenesis. Okay, and you obtain that from alanine here. Okay, so you can see, now you're seeing pyruvate, which is this one here. Okay, so in the case, if this was alanine, right, we're giving the amino group to keto acid number 1, which is this one here. And then the keto acid number 2 is pyruvate. And then tyrosine will be the new, glutamate will be the new amino acid. Okay, so you can see what's going on here. Okay, so so this is the transamination of alanine to make pyruvate and glutamate. Now, here on the other one here, you can see aspartate. Okay, aspartate is another amino acid. So here's the amino group from aspartate. It's going to be transferred to paradoxal phosphate. It's going to become paradoxamine phosphate, then again, this is mediated by this transaminase here, aspartate transferase. And then it comes again here, aspartate transferase again. And it's going to mediate the transfer of this amino group to this keto acid number 1. And you're going to generate glutamate again. Look at that, glutamate, which is going to be available for protein synthesis. So the new amino acid is going to be available for protein synthesis. And the keto acid number 2 can be used for gluconeogenesis. Oxaloacetate is an important intermediate that we see in the Krebs cycle. In the Krebs cycle, remember that in carbohydrate metabolism, when pyruvate got into the Krebs cycle, got into the matrix of the mitochondria, then it was converted to acetyl-CoA. Acetyl-CoA combines with oxaloacetate, and we can go into the Krebs cycle and make NADHs and FADHs and make energy from it. So you can see that the body has an excellent, it's great in recycling things. This is another takeaway from this. By the way, these processes of transamination and deamination, the amination occurs primarily in the mitochondria, and transamination can occur in both. We have enzymes that participate in transamination that come from the mitochondria, and the things that are taking place in the mitochondria, or the things that are taking place in the cytoplasm. Another reason to love your mom. That your mom is the one that gave you the mitochondria. It wasn't your dad. Your dad didn't give you mitochondria, your mom did. So now you learn about that the mitochondria is the powerhouse of the cell, because it's where most of the energy is made. And look at this. This is where transamination and deamination reactions are taking place as well, which are very crucial to keep everything, so you can recycle. When you have an excess of nitrogen, you can get rid of it in the form of ammonium ions, but you can also recycle nitrogen. You can also recycle that and make other amino acids and other products here, other amino acids, I should say. Okay, so let's keep going here. So you can see that transaminase enzymes are important in the production of various amino acids, and in measuring the concentrations of various transaminases in the blood is important in the diagnosing and tracking many diseases. Okay, for example, the presence of elevated transaminases can be an indicator of liver and cardiac damage. Like, people that have heart failure, myocardial infarction, you heard about that they were, your cardiac enzymes are elevated. One of the cardiac enzymes that can be elevated is aspartate transferase. Aspartate transaminase, or aminotransferase, so aspartate transaminase. This one here, AST, can be elevated in people that have, a myocardial infarction. It's non-specific, and it takes a while for it to appear. So now they're taking troponin as the major marker for myocardial infarction. Okay, but myocardial infarction it's confirmed by doing a blood test and testing for cardiac enzymes being elevated, or troponin levels. Okay. So two important transaminase enzymes are aspartate transaminase, also known as serum glutamic oxaloacetic transaminase. That was an older term, Now we use these newer terms here: aspartate transaminase and alanine transaminase. We'll keep it as that. AST, ALT. ALT is more specific on liver failure. AST is more, is broad. It's not as specific as ALT. Now, elevated liver enzymes, okay, notice it says here, as well as in liver tissue, AST is also present in cardiac. So you see AST, aspartate transaminase is not only present in liver, but also is present in cardiac and skeletal muscle, and even in red blood cells. But ALT, alanine transaminase, that one is the most specific marker for liver damage. Okay, so when they do a liver function test, they're testing for levels of AST and ALT, specifically ALT. Okay, that's why I put in I should have put an asterisk over here, there, instead of here, because we're gonna keep the easier name to remember. Okay, let's move on. Here's oxidative deamination. Oxidative deamination. Okay. Now notice that we have glutamate here. Okay, and we need a deaminase, an enzyme here, to do this process. So in this case, we're going to need glutamate dehydrogenase. That will be the deaminase that is going to carry this reaction here. And notice that, we're generating NADH. This is an oxidative process, by the way. This is an oxidizing agent here. So we're generating energy. And you can see that we're generating a keto acid, okay? And that will be the carbon backbone. We'll keep the carbon backbone. So we can suspect that this is, again, used for energy. And then ammonium ions will be a toxic, a waste product that we need to get rid of because it's toxic to your brain. People with liver failure, would have increased amounts of ammonium ions, of ammonia, for example, and they will feel confused, and eventually they'll die. Okay, so usually people are put into hospice, when that happens, because ammonium, the liver, there is something called the urea cycle that happens in your liver. Okay, that happens actually in the mitochondria of hepatocytes. And that's where ammonium ions are converted into urea. They're combined with, ammonium ions are combined with carbon dioxide. With carbon dioxide. You'll see it here, the urea cycle, in a moment. Okay, so you can see that ammonium ions are combined with carbon dioxide. Okay, and we're getting urea. So urea is less toxic than ammonia. And that's how we want to release it into the blood. Okay, so I'll tell you a story. This is interesting, okay? And this is why I emphasize in the importance of knowing the details, knowing, the information well, because it can be handy to make wise decisions, because you cannot rely on other people. Unfortunately, not everybody's the same, and nobody is as good as you. Okay, so not that you have to be arrogant or anything like that, but the more knowledgeable you are, the more power you have, and the more the better decisions you're going to make. Okay, so for example, I was talking to my aunt. My aunt had diabetes for a long time. Okay, and I was talking to her. She was in rehab. She was doing very well. I went to visit her, and then I was talking to her normally. There was no signs of confusion or anything like that. I don't know what happened, but a week later, I found out that she's in hospice. And I'm like, saying, what happened here? But I found out like 3 days into hospice. So in 3 days after she was in hospice, so she was already being administered, morphine for her comfort and pain, as they say. And they stopped treating her for diabetes or anything like that, because, she was, that's it, she was, her husband was told that she needed to go to hospice. And that was the end of her life. I asked her husband, what happened? And he tells me, the doctors said to me that she has an incurable bacteria. There is a bacteria that there is no treatment for it. And he said, what's the name of the bacteria? He said, ammonia. I said, ammonia is not a bacteria. Ammonia is a chemical. It's a toxic chemical made by the liver, during the deamination process here. It's a way to keep nitrogen balance, in your body. And if you have too much nitrogen, you need to get rid of it, right? But you can also recycle nitrogen through transamination, okay? Because you can fabricate another amino acid, right? So I said, that's not a bacteria, that's a chemical, that's ammonia. And how do you make ammonia? When you have an excess of nitrogen. How do you get an excess of nitrogen? From consuming too many proteins. So she was giving a lot of proteins, perhaps, and her liver was not able to convert these ammonium ions into, into urea. So in that case, what they needed to do was to restrict her from proteins, or cut down on the number of proteins. So the liver, which has the capacity to regenerate itself, hopefully she recovered from it, But I guess they didn't want to deal with her. I don't know what that was, but she was very young, and it's very unfortunate, especially when he told me that ammonia was a bacteria. He didn't have an understanding, and nor did they call me. I don't understand, why they didn't call me, but I want to show you something else about deamination. Okay, thanks to somebody online who is a biochemist, and he has excellent demonstrations, all credit to him. Okay. You can see here with more clarity, okay, not just these simple images here. What's going on here? So you can see that glutamate, by the way, glutamate is a predominant amino acid in the blood, glutamate, okay? And you can see that glutamate can take another amino group, okay? And there's an enzyme here that, that is needed for this to happen. And since we're building, we need ATP, right? So now we added an extra amino group here that we can get rid of, okay? So let's see what happens here. Okay, so here is glutamine. Now we have glutamine. Now we had glutamate, but once we added the extra amino group, now it's called glutamine. And glutamine, in the presence of this enzyme, which is called glutaminase, we're gonna get rid of those extra amino groups here in the form of ammonia. And then we're back to glutamate again. We're recycling it again. But then glutamate, we can get rid of more of the other amino group here, right? And then now we get alpha ketoglutarate. Wow. Alpha ketoglutarate was that second keto acid that we saw prior, right here. Alpha ketoglutarate, right? You can see that. And then we can, we can transfer amino groups for their elimination if we have an excess of them. How beautiful is this, So that's when you dig into it. I had a professor, her name was, I don't remember her name, but she was an infectious disease doctor, okay, an infectious disease doctor. And she said to us, that who looks will find. That who looks will find the answer. The purpose of learning, the purpose is not just memorization, but also, is to learn how things are applied, how they're useful, that is learn with purpose, just don't just memorize, learn with purpose, okay? And so, this is like my third recording. I needed to go back and do another, this recording here to be more effective in what I wanted to say and transmit here, okay? But notice that, the transamination reactions here and the amination reactions can take place here in both of these organs, okay? Usually transaminations, you refer to as liver. There is also, transamination can also happen in your muscles as well, okay? And that's why, I found another image that I have at the end of the notes here to show you that. But again, it's a lot, it's a lot of information. It's a lot of research. It took me a whole day to research this information very well, because I wanted to give you the best of me, okay? So, let's see what else from here. Here's the, for protein synthesis, we need amino acids, and these amino acids can be made by your body to transamination, okay? And other ones you can get from your diet. And the ones you get from your diet are the essential amino acids. So in reality, there's 8 of them, 8 that are not, that you get from your diet. But I usually tell you 10 of them, that there's 10, because you can see here that 2 can be insufficiently made, which are arginine and histidine, and they're insufficient for growing children, okay? So, and by the way, these are mnemonic here to remember the essential amino acids, like phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. Okay, so that's just to remember. PBT Tim Hall. Okay, that's from medical school. Okay. So I explain here how, these other amino acids can perhaps not be made in, that perhaps your body might run out of them, not be made sufficiently by your body. So for example, it is possible for vegans to consume all 9 essential amino acids. Sometimes they tell you 9, sometimes they tell you 8, sometimes they tell you 10, In this case, they're referring to 9, okay? So there is one of them here. So for example, grains like rice are too low in lysine to be a complete source of protein. But because lentils and beans are higher in lysine, you can obtain all 9 essential amino acids by eating them with grains. We're talking about vegans here. Okay, so in vegans, lysine might be insufficiently made by your body, right? But if they complement it with other things, and not just rice, okay, you can get the other amino acid. Now, let's talk about, you see like arginine and histidine here. So arginine is considered a non-essential amino acid in healthy adults because the body can synthesize enough of it to maintain muscle mass and connective tissue. However, arginine can be conditionally essential in certain situations, such as during growth, pregnancy. People, women that are pregnant, they can have a deficiency in arginine. There is a, children that are growing, okay? Trauma, infection, when the body's production capacity is exceeded, okay? In these cases, arginine must be obtained from, through the diet, because the body is not, it's not producing enough to deal with these problems here. Arginine is also conditionally essential for infants and growing children. So it can be essential for infants and growing children, as well as adults with kidney or small intestine dysfunctions. So in your children, are they getting enough arginine? Look for a diet that has arginine, because they might be deficient, it's needed for growth. Okay, so that's important. Okay, and then in here, I gathered some images, some extra images. You can look at them. But one is for lipid metabolism, and the other one here to show you that transamination can also happen in muscle. You can see that amino acids, amino groups can be transferred from amino acids to pyruvate, and then you get alanine, and alanine goes into the liver, and it undergoes this transamination. All right. So how'd you like that? All right, so that's it. That's it for this presentation. I hope you liked it. I hope you have a better understanding of this information. At the end, learn with purpose, please. Don't just take somebody's word for it. Don't just memorize things. No, really understand what's going on so you can make better decisions for you and for your loved ones, for somebody else that you in the health field. I see everybody as my family. Okay, so that's it. So see you in a future recording. Okay, bye-bye.